There's a whole universe of machines and molecules inside us that we don't appreciate enough. They keep us alive after all. Here on Trouble Helix, we acknowledge these inconceivable yet essential little guys who shape our very existence. I'm Rebecca Ravenstad, and I'll be your existential crisis counselor. My credentials. I have a bachelor's in molecular, cell, and developmental biology, the big three. I worked in both biotech and research, and while that was super fun, I much prefer spreading the gospel of the most compelling branches of biology. Each episode will be an absolute revelation about the molecules that fuel our continued survival, the mystery behind diseases, bodily functions, and maybe even life itself will crumble before you. Whew, pardon me. Today, we will be exploring a molecular mishap in which the absence of a single molecule causes salt wasting and sex reversal. Congenital Adrenal Hyperplasia, or CAH, is a genetic disease of the adrenal glands. These triangular little hats for our kidneys synthesize and release hormones. Famously, adrenaline is released by the inner medulla, while steroid hormones are released by the outer cortex. In CAH, the cortex is busted, and in 95% of cases, it's because of a deficiency in one enzyme, 21-hydroxylase. This enzyme is responsible for making two of the steroid hormones, aldosterone and cortisol. Life-threatening symptoms of CAH, such as low blood volume, metabolic acidosis, and low blood sugar, occur due to the lack of those hormones. Sex-related symptoms like ambiguous genitalia and reduced fertility occur due to the imbalances that arise in the steroid hormone pathway. To understand why it's so devastating to lose this one enzyme, I'm going to show you what's supposed to go on in the adrenal cortex, how we're supposed to make the steroid hormones. I'll show you what aldosterone and cortisol do for us and explain why it's dangerous to not have them around. I'll even give you the sex talk. To begin our journey, let's go back to where this all began, the eggs and sperm of your parents. A lot of times, molecular mishaps occur due to tiny mistakes that take place well before the patient is even born. It happens during the formation of the parents' sperm and egg cells, their gametes. This is when genes that'll eventually be passed down to their offspring are being reorganized and carefully placed into their own cells. This is meiosis. During meiosis, our gene libraries, or chromosomes, replicate and then touch each other, exchanging genetic material in a process called crossing over. Ideally, the same chromosomes line up and cross over with each other, with their homologs. So chromosome 1 from the egg parent recombines with chromosome 1 of the sperm parent. This happens for all 23 different chromosomes in order to create genetically diverse gametes. It's astonishing that this can all take place without a hitch. But sometimes, a dumb hitch will show itself in the form of an imposter gene. CYP21A2 is the gene that codes for our protein 21-hydroxylase, and it's located on chromosome 6. Also, for some reason, there's a nearly identical pseudogene called CYP21A2P located practically right next to it. We'll call that gene dumb P. When the two chromosome 6s meet up during meiosis, CYP21A2 can wind up interacting with dumb P on its homolog instead of the other CYP21A2. This is such a dumb mistake, but they're so alike that it reads as a correct alignment. This has some serious consequences because dumb P is totally useless and encodes nothing. Instead of exchanging information within the same gene to create variation, dumb P is poisoning the healthy gene with its messed up sequences. Now, when meiosis continues and the chromosomes segregate into separate gametes, some of those cells will have a chromosome 6 with a ruined CYP21A2 gene. Most of the time, It will actually get completely replaced by DUMP, but sometimes a worthless freak chimera of the two genes can result. If an embryo develops from both an egg and a sperm containing ruined CYP21A2 genes, then it will have 21-hydroxylase deficiency and therefore suffer from CAH. So, a meiotic miscalculation knocks out our star enzyme and leads to dysfunction. But let's get physiological before we get pathological. How does 21-hydroxylase contribute to our overall health? Steroid hormones are made from cholesterol via steroidogenesis. A molecule of cholesterol will undergo a series of chemical changes and transform into one of several classes of steroid hormones: androgens, estrogens, glucocorticoids like cortisol, or mineralocorticoids like aldosterone. The fate of the cholesterol depends on the enzymes being expressed in a given tissue. For example, androgens are specifically made in a layer of the adrenal cortex called the zona reticularis. To make an androgen, we need to allow cholesterol into the cells that make up the zona reticularis, and then we need to let it into the mitochondria of those cells. This is where steroidogenesis begins. Viewers will get a nice, organized representation of the pathway, and listeners, get ready for some words. The first thing we need to do once we've trapped a molecule of cholesterol is chop off its arm and turn it into pregnenolone. Pregnenolone is the pillar of our steroidogenic choose-your-own-adventure. It can interact with multiple enzymes and become pretty much any of the steroid hormones. But because we are in the zona reticularis right now, the enzyme 17-alpha-hydroxylase is going to be the one to convert it into 17-alpha-hydroxypregnenolone. Easy. We're just adding a hydroxyl group, an OH, to the 17th carbon of pregnenolone. It's in the name. Then we just have one more step. 1720-lyase is going to turn that into dehydroepiandrosterone, or DHEA. This is the main androgen that the adrenals produce, and its function is to get turned into other things. In the testes, it gets converted into testosterone, a classic, which can then be converted into the strongest of the androgens, dihydrotestosterone, or DHT. The adrenals can't make their own DHT because they don't express the enzyme 5-alpha reductase, so they provide a supply of DHEA that can be transformed into stronger androgens by other tissues. Androgens can also be converted into, plot twist, the estrogens. They both mediate certain behavioral and sexual characteristics, and their effects differ depending on your sex. That's why they're called the sex hormones. DHEA becomes the estrogens in the ovaries because they express the enzyme aromatase, which makes estradiol from testosterone. Already, you should be clued in to the intimacy of these molecular interactions, despite 21-hydroxylase's lack of direct role in sex hormone production. Conversely, 21-hydroxylase is essential in the production of glucocorticoids. Cortisol is made in the zona fasciculata of the adrenal cortex, and although we're still going to start with pregnenolone, the path it takes will be different. Unlike the zona reticularis, the fasciculata has a high amount of an enzyme called 3-beta-hydroxysteroid dehydrogenase, which converts pregnenolone into progesterone and 17-hydroxypregnenolone into 17-hydroxyprogesterone. This effectively shifts steroidogenesis away from the androgens and provides substrates for 21-hydroxylase. To make cortisol, 21-hydroxylase will convert 17-hydroxyprogesterone into 11-deoxycortisol. And finally, 11-beta-hydroxylase will convert that into cortisol. Note that because the fasciculata and reticularis don't express the same levels of the same enzymes, they're somewhat specialized in the steroid hormones they make. The reticularis can't really make cortisol because it lacks 3-beta-HSD, and the fasciculata can't really make androgens because of its low 1720-lyase activity. The final, outer layer of the adrenal cortex is the most specialized. It can make something that no other cells of the body can make. The zona glomerulosa makes aldosterone. Here, our pregnenolone must be converted to progesterone with 3-beta-HSD. There's almost no 17-alpha hydroxylase here, so the creation of androgens or cortisol is unlikely. This time, progesterone will be the lucky recipient of an OH group from 21-hydroxylase, turning it into deoxycorticosterone. 11-beta-hydroxylase will make another appearance to turn deoxycorticosterone into plain old corticosterone. We're almost there. This final step is what makes the zona glomerulosa the most unique layer of the adrenal cortex. It's the only area in the body that has aldosterone synthase, which turns corticosterone into aldosterone. Voila! The steroid hormones. What a journey! I want you to really understand that the reason tissues and organs and cells can specialize is thanks to distinct patterns of protein expression. That's why, for example, the reticularis can't produce aldosterone. All of our cells contain the recipe, the gene for making aldosterone synthase, but it's only the cells of the glomerulosa that can make it because that gene is turned off everywhere else. This fundamental molecular concept is called epigenetics. All cells have the same code, but different parts of that code may be turned on or off to create different cell identities. In going through the steroidogenic pathway, we've seen that 21-hydroxylase sits at a pivotal position in the production of steroids. The extent of the slack created in its absence is becoming more evident. What do pregnenolone, 17-hydroxypregnenolone, progesterone, and 17-hydroxyprogesterone all have in common? That's right, they can all be shuttled into the pathway that leads directly to the androgens, causing an overproduction of those hormones at the expense of some other members of the steroid family. As we begin our descent from physiological to pathological, let's see how aldosterone and cortisol help us not die. Blood pressure is incredibly important. So much so that our body employs multiple methods to ensure that it stays within physiological range. Constricting or dilating our blood vessels is a rapid approach to blood pressure regulation, but long-term maintenance calls for an inside job. Aldosterone alters the contents of our blood vessels. It harnesses the magic of the electrochemical gradient to adjust our blood volume, which is directly related to our blood pressure. Having more liquid in our tubes means more pressure being exerted from within. And although aldosterone's ultimate goal is to keep our electrolytes balanced, this function is critical to the maintenance of our blood pressure. In fact, aldosterone is a member of the renin-angiotensin-aldosterone system, or RAS, a process that causes a global increase in blood pressure. It involves a chain of enzymes and hormones that constricts the cardio system, increases heart rate, and retains water, all as a way to bring blood pressure back to normal. Our body doesn't necessarily know the specific cause of low blood pressure, so it just sounds all of the alarms by activating RAS. We just care about the alarm that goes off in the kidneys, though, the one that summons aldosterone to correct any electrolyte and blood volume issues. Drops in blood pressure will be detected by juxtaglomerular cells in our kidneys, which sets off RAS and leads to the formation of angiotensin II. This guy will both constrict blood vessels and stimulate aldosterone secretion from the zona glomerulosa. Now, in addition to squeezing our blood tubes, it's time for us to refill them by changing what the kidneys reabsorb back into our body and what they excrete in our urine. Inside the kidneys are millions of nephrons, tiny structures that filter our blood to make urine, which then carries waste out of our body. The last parts of the nephron, the distal convoluted tubule and the collecting duct, contain fluid that is more urine than blood. This is where aldosterone will work its magic. It's going to change their permeability to sodium and potassium in a fascinating way. Aldosterone, using its steroid hormone privilege, will diffuse directly into the cell where it will find its mineralocorticoid receptor, or MR. The MR is a nuclear receptor, so when aldosterone binds, the whole complex is going to go into the nucleus and bind directly to a specific section of DNA called a hormone response element. This tells the cell that it needs to start transcribing the gene indicated by the location of the HRE and churn out the protein encoded by that gene. In this case, aldosterone tells the cell to start making two proteins, ENaC and the sodium-potassium ATPase. Let's walk through that one more time. Aldosterone forces its way into the very blueprints of your existence and says, make these two proteins so that we don't die. This is another fundamental concept of molecular biology called gene regulation, where genes are turned on or off depending on our physiological needs. Amazing! These two proteins are going to help the cells of the DCT and collecting duct reabsorb sodium and excrete potassium. ENaC will insert itself into the cell membrane on the luminal side, the side that faces the lumen, or the inside of the tubule. It's going to take in sodium from that tubular fluid that would otherwise be urinated away and bring it into the cell. On the other side, the sodium-potassium ATPase is going to pump that same sodium out of the cell and into the extracellular fluid, which includes our blood plasma. The complete opposite will happen with potassium. In exchange for the incoming sodium, potassium from the ECF will enter the cell, get pushed into the tubular fluid, and eventually excreted. As more sodium is being pumped into our ECF than remains within our cells, osmotic pressure is created and water will follow the sodium. Water is drawn to areas that have a higher solute concentration. With more liquid being reabsorbed, blood volume will rise, and so will blood pressure. Balance. RAS is an important stimulator of aldosterone secretion, but it's not the only one, or even the fastest one. We can skip the R and the A and rapidly trigger aldosterone with hyperkalemia, high blood potassium. This will increase the amount of those sodium-potassium ATPases, which will help get that excess potassium out of the body. This is another important point of regulation besides blood pressure because we want our blood to be at a normal volume, but we want the contents of our blood, cells, and the rest of the fluids in our body to be electrochemically balanced. Sodium and potassium are both positive ions, so their exchange helps maintain this electrochemical equality, but potassium isn't like other girls. Most ions are more concentrated outside versus inside the cell, but potassium is much more concentrated inside of cells. This disparity is vital for heart and nerve function because these systems use electrical signals to communicate and coordinate muscular functions, and they rely on changes in charge across a membrane in order to send those signals. Balance. The last ion that aldosterone supervises is H+, aka protons, aka acid. It helps us excrete acid by increasing the expression of a proton pump that pumps acid out of the cell and into the lumen. Also, aldosterone's promotion of sodium uptake creates a gradient. It makes the inside of the cell more positive than the lumen, and this drives the movement of acid into the lumen where it's less positive so that it can be excreted. Balance. Aldosterone does all these amazing things to maintain balance within our body, unless, of course, you have 21-hydroxylase deficiency. Without aldosterone, blood pressure regulation will be devoid of any volume regulation, and the patient may suffer from hypovolemia or low blood volume, potentially deadly. Without aldosterone, Potassium won't be excreted efficiently, and the patient may suffer from hyperkalemia. This disrupts our body's electrical signaling, leading to neuromuscular impairments and arrhythmia, potentially deadly. Inefficient acid secretion may cause metabolic acidosis, which is potentially deadly. Newborns with CAH must be attended to quickly before they suffer from any of these fatal symptoms. Luckily, in the US at least, screening for CAH is done a few days after birth so that care can potentially start before the symptoms do. IV electrolytes reverse the negative effects, as does the administration of hydrocortisone. With all the critical operations that aldosterone performs for us, it's easy to forget about cortisol. Aldosterone makes my blood correct. What could be more important than that? Do we really even need cortisol? We call it the stress hormone for a reason. All it does is make me stressed. Maybe we'd all be better without cortisol. Okay, cortisol gets a bad rap, but I promise it's just misunderstood. It's not called the stress hormone because it makes us stressed. It's because cortisol helps us deal with it. Stress doesn't just mean running from a predator, a common example of something that requires a lot of energy. Humans are apex predators, so we don't really experience that, but cortisol is still necessary for any other stressor that you or I may encounter. Taking a calc exam, being too hot, being too cold, doing drugs, meeting societal expectations, being sick. All of those are stressors that may require some level of energy to overcome. Cortisol helps us deal by releasing our stored energy in the form of glucose and fatty acids. Cortisol is all about energy mobilization. It tells our liver to make a ton of glucose at the cost of our skeletal muscle. We need the proteins in our muscles to get broken down into amino acids, which the liver then uses to generate glucose. Glucose is your brain's favorite source of energy, and since the brain is so damn important, we want to make sure that other parts of the body aren't tapping into that supply. Cortisol prevents muscle, fat, and even the liver itself from taking in glucose, meaning another source of energy must be used. Fatty acids release a lot of energy when used, and cortisol acts on fat cells to break down triglycerides into fatty acids and glycerol. Now the liver can utilize this form of energy in order to continue gluconeogenesis. Mobilization of glucose and fatty acids provides our body with energy that can be expended to make more energy. Of course, it can also be expended to fuel a stressful situation. Taking an exam can require a significant amount of energy from the brain, justifying the need for increased glucose production. Cortisol also functions to anticipate and prepare for stressors. Although it calls for the liver to make glucose, it also instructs the liver to store more glucose in the form of glycogen. This prevents the body from depleting itself of an energy source and keeps itself prepared in case it becomes starved, for example. Cortisol is just looking out for you. As a member of an endocrine pathway called the Hypothalamic-Pituitary-Adrenal Axis, or HPA axis, cortisol is also looking out for itself. The whole point of the endocrine system, the whole point of hormones, is to communicate with distant areas of the body to coordinate and regulate the majority of our bodily functions. Hence, signals from our brain, for example, can trigger hormone release elsewhere. The hypothalamus is a homeostatic control center and stress sensor within our brain. In response to stressful triggers, it will secrete a hormone called corticotropin-releasing hormone. CRH then acts on the pituitary gland to stimulate the release of adrenocorticotropic hormone, or ACTH. From the brain, ACTH will go to the adrenal glands and stimulate the release of cortisol. At this point, cortisol will go about its day and carry out its functions, one of which is regulating the HPA axis. Cortisol completes the axis by exerting negative feedback on the hypothalamus and pituitary, meaning that the more cortisol we have in our system, the less CRH and ACTH will be released. The adrenals are essentially letting the brain know that there's enough cortisol circulating that it no longer needs to be stimulated. Low CRH and ACTH means eventually cortisol levels will drop and the cycle can continue. Without enough cortisol to inhibit the hypothalamus and pituitary, they'll resume their hormone secretion and stimulate more cortisol. This is a classic endocrine pathway where the signals originating in the brain are subject to negative feedback by the effector organ they stimulate, in this case, the adrenals. So, what happens when we remove the key regulator of this pathway? Big problems. No cortisol means there's nothing to keep CRH and ACTH in check, so they just run rampant. The unsupervised hypothalamus will constantly demand that the pituitary release ACTH, and this hormone's got its own agenda. It wants to increase steroidogenesis any way it can. Since cholesterol is necessary for steroid hormone production, ACTH increases the expression of low-density lipoprotein receptors on the cells. LDL is a protein that transports cholesterol throughout our bloodstream, so more of its receptors means more cholesterol being shuttled into the cell. Steroidogenesis actually takes place in the mitochondrion, though, but don't worry. ACTH also raises the expression of steroidogenic acute regulatory protein, or STAR. This guy grabs cholesterol from the cytosol and brings it into the mitochondrion. More cholesterol being brought into the cell is certainly enough to generate more steroids, but ACTH doesn't stop there. It also boosts the activity and expression of all the enzymes in the pathway that lead to cortisol. Oh, and I should mention, this isn't just happening in the zona fasciculata. ACTH is also dominating the cells of the zona reticularis, where androgens are produced. As ACTH continues to overwork the cells of the adrenal glands, they will multiply at a pathological rate and the glands will swell in size. This is the H in CAH, hyperplasia. Boosting the steroidogenic enzymes without the presence of 21-hydroxylase will cause a buildup of precursors to cortisol, and those precursors are going to shack up with the other available enzymes in the pathway. The adrenals now sit atop the kidneys as two androgen factories, reluctant yet robust in their mission to revise the course of human development, regardless of the genetic guidelines. Oops, all androgens. Everyone's got them, but no one is supposed to have this much of them. When a newborn is suspected to have CAH, one of the markers they'll test for is 17-alpha-hydroxyprogesterone. In CAH, this molecule is elevated because it can't be converted into 11-deoxycortisol without 21-hydroxylase. So instead, 17-OHP will go down the androgen rabbit hole. 17-OHP, and also progesterone, can be converted into testosterone via the classic pathway, but they can also be shunted into the androgen backdoor pathway. Normally, testosterone will undergo a 5-alpha reduction to become DHT in the testes. If we take the backdoor pathway, we can bypass the creation of testosterone and get to DHT by performing a 5-alpha reduction on 17-OHP and progesterone. This roundabout path also involves the production of a host of other androgens, all being promoted by our good friend, ACTH. When the appropriate amount of androgens is being made, they regulate the development of genitalia, specifically the testes and prostate. They regulate skin maturation, muscle mass, certain behaviors, and fertility. Everyone has skin and muscle and behaviors, so you can assume that everyone produces androgens. In fact, they're pretty much always present in higher amounts than estrogens, no matter who you are. But since they're sex hormones, they're gonna do different things to different people. I feel like I've grown a lot throughout this discussion, so I think I'm finally ready to bite the bullet and give you the sex talk. There are at least 4 types of sex in the world. Say 'em with me: chromosomal, gonadal, hormonal, and anatomical. Depending on the developmental intention of a fetus, CAH can create disparities between the four sexes. Sex chromosomes determine which path, which gonads are going to be made, and then the ratio of hormones secreted by those gonads informs the characteristics associated with male or female anatomy. Do they always line up perfectly? Of course not. But regardless of their alignment, and regardless of how society views the final product, there are two configurations encoded in our DNA that we can arrive at from two different sexual development paths: testes or ovaries, sperm or egg, Sertoli and Leydig cells or granulosa and theca cells, Wolffian ducts or Mullerian ducts, male configuration or female configuration. The parts, not the person.
We're not talking about this or this. We're talking about this.
Okay, so sex reversal is exactly what it sounds like. It's when one developmental direction is being taken and then something completely changes the course of development despite the path that was determined by the sex chromosomes. On its own, it's not pathological. Sex reversal and ambiguous genitalia don't always present with clear disease conditions, though they are stigmatized. 21-hydroxylase deficiency is a disease, though, and its effects on sexual development are pathological. I'm going to walk you through human sex determination and development, something my own development professor didn't even do because it was too complicated, and then we'll reverse it. Newborns that are slated to enter the world with the female configuration can have their sex reversed by CAH in a process called virilization. This takes place in utero because the overproduction of androgens by the fetus is interfering with other signals, with the expression of certain genes that are needed for proper female development. Let's all think way back to when we were all fetuses around our 6th week of gestation. If you're feeling a bit weird, it's because you have all the genitals, or rather, you have the tools to make any genitalia. We have a bipotential gonad, and it has all the tubes necessary for making whatever reproductive system it wants. We've got Mullerian ducts in case we want to make some ovaries, and Wolffian ducts in case we want to make some testes. It has yet to receive molecular signals telling it to become one thing or another. After the 6th week, a combination of genetic, molecular, and cellular factors will start tipping the scales. If a Y chromosome is present, then so is the SRY gene, which encodes a protein called testis determining factor. This protein initiates and orchestrates male development and will activate some other genes that aren't even on the sex chromosomes, like FGF9, which is on chromosome 13, and SOX9, which is on chromosome 17. Activation of those genes, and therefore production of their proteins, is what sparks the development of Sertoli cells during week 8. These are one of the two main cells in the testes, and their first order of business is to release anti-Mullerian hormone, or AMH. This deletes those Mullerian ducts because we're not going to be making any ovaries. The cells of the bipotential gonad will continue to differentiate, and another testis-specific cell will arise, the Leydig cells, which produce steroid hormones. Eventually, their presence will cause a spike in testosterone, which helps to differentiate the Wolffian ducts into internal male reproductive structures. To reiterate, the typical chromosomal sex is XY. The SRY gene on the Y chromosome specifies the gonadal sex by directing other proteins required for male development to help differentiate the bipotential gonad into testes. The cell types unique to the testes will make hormones that are needed for male development. If all goes well, the gonads, as well as all of the other structures of the reproductive system, will be anatomically correct. Boom! All sexes present and accounted for. Lack of a Y chromosome is what determines female sex, and the regular style chromosome combination is XX. Without SRY, female development may seem like the default path, but in reality, It's an active process that still calls for the coordination of female sex genes and the suppression of male sex genes. SRY may be the master sex development gene, but SOX9 expression without SRY is enough to stimulate testis development. It truly takes a village. So, during female development, genes like FOXL2 and respondin-1 will repress any genes that promote male development, and vice versa. Female promoting genes will be suppressed in males. An incredibly important gene in female development, WENT4, makes sure that the cells of the bipotential gonad lean toward ovarian cells rather than becoming testosterone-producing Leydig cells. The absence of androgens will cause the Wolffian ducts to degrade, and the Mullerian ducts will continue developing into the female reproductive system. By week 10 or so, the internal reproductive system from either path is complete. By this point, circulating fetal androgens and estrogens have all come from the cells of their respective gonads. It isn't until week 12 that the fetal adrenal glands can contribute to steroid hormone production, so an XX individual will have already developed their internal reproductive structures: ovaries, oviducts, and a uterus. These adrenal glands, though, are 21-hydroxylase deficient, so androgen levels will quickly skyrocket as the glands fail to produce aldosterone and cortisol. These unexpected and excessive androgens will make anatomical changes to the external reproductive structures, so the clitoris may resemble a penis, the labia may resemble a scrotum, and the vagina may be closed. Again, this is only the external genitalia, so the scrotum will be empty as the ovaries have already been formed. Let's review the sexes. The chromosomal sex, XX, was consistent with the expected gonadal sex, ovaries. But the hormonal sex that arose after gonad formation affected the anatomical sex such that a more male form resulted. I said before that while ambiguous genitalia is not inherently pathological, it could be in the case of CAH. This has less to do with the level of virilization, which can range from mild to complete, and more to do with the complications that arise if you have conflicting reproductive structures. For example, penises and vaginas both have urethras, but in completely different places. So, if this confusion happened where male external genitalia was incompletely created, that urethra might be in the wrong place. Not life or death, but potentially problematic. Sometimes the urethra might connect to the bladder through the upper vagina and uterus, which could have medical consequences. Androgens continue to function well after birth, so their aberrant production in CAH can extend into puberty and adulthood. You've heard of the HPA axis. Now let's look at the HPG axis, an endocrine pathway that involves our gonads. Remember that cortisol prevents our hypothalamus from releasing CRH and our pituitary from releasing ACTH. Androgens also exert negative feedback on those areas to prevent the secretion of gonadotropin-releasing hormone from the hypothalamus and the gonadotropins from the pituitary. With the HPA axis, we saw how no negative feedback affected the body, but with the HPG axis, too much negative feedback screws things up. The gonadotropins, follicle-stimulating hormone and luteinizing hormone, are important for all the gonads. They work synergistically to support sexual health, like maintaining spermatogenesis and regulating the menstrual cycle. They also promote the production of sex hormones, affecting secondary sex characteristics like hair growth and fat distribution. If poorly managed, the androgen excess from CAH can cause fertility and sex-related issues in males and females, generally due to the suppression of the HPG axis. A person with ovaries might have an irregular or absent period without the synergy of FSH and LH. There may also be other androgen-coded characteristics besides ambiguous genitalia, like excess hair growth, acne, and early puberty. Males with CAH are also subject to these hypermasculine characteristics as well as lower rates of fertility. In fact, the size of the testes is decreased in CAH due to the lack of gonadotropins. Hyperandrogenism inhibits normal gonadal health and fertility by bombarding the HPG axis with negative feedback and by exerting its own direct effects on the body. Managing the androgen excess of CAH can be done with hormone replacement, where the steroids that the body is supposed to be making are taken as medicine. Hydrocortisone to make up for the absent cortisol will also curb the production of androgens since it will restore negative feedback to the HPA axis. To replace aldosterone, a medication called fludrocortisone helps the patient retain sodium and blood volume. This whole disease came about because a perfectly functional gene was tricked by its evil twin. A mishap that was powerful enough to reverse the course of sexual development, to reverse the path of steroidogenesis. It truly baffles me how we can even get past fertilization and put all 20,000 of our genes to work. Thank you very much for listening. I hope you enjoyed yourself, and I hope you learned a little something about the insanity that resides within us. Everything you heard was researched, recorded, written, edited, and composed by me, Rebecca Ravenstad. If you're looking for more trouble, you can find the website for my sources on whichever service you're currently using. Again, I greatly appreciate you listening and encourage you to join me next time for more molecules and mishaps.